Bonjour Jackie, hello everybody. So yes, I'm a first year CTN postdoc under the supervision of Dr. Jean-Pierre Gutsy in Montreal. And today I'll present two studies we are developing with the support of the CTN, as Jackie said, the Kamikami study and the Gutsy study. So I have no conflict of interest. So we know that in Canada, most people living with HIV have access to antiretroviral therapy. They are successfully treated and have low to undetectable viremia. And the life expectancy is similar to the one of the general population. But we know that those people, as we heard before today and yesterday, that they want to age in health. They want to be able to age without comorbidities, because we know that they have increased risk of non-AIDS comorbidities, such as cardiovascular, bone diseases, metabolic disease, and cancer. As we heard also before, the main driver of those, it's actually persisting inflammation that is responsible for those comorbidities, even after years of antiretroviral therapy. There are several mechanisms that are linked to drive persisting inflammation. And one of the main recognized mechanisms nowadays is actually the leaky gut. So it's the gut epithelium that allows the passage of microbes or microproducts in the body, inducing inflammation, and it's a process called microbial translocation. So we also know that the gut microbiota, meaning the microbes that naturally live in our gut, have a different composition in people living with HIV and the antiretroviral therapy compared to negative controls. And people living with HIV have more pro-inflammatory microbes and less beneficial microbes. And actually, the composition of this microbiota has been linked to the persisting inflammation under heart. So this different composition has recently been linked, or is also called dysbiosis. So the gut is different, the gut microbiota is different. And this dysbiosis has been linked with the development of comorbidities. So we need to develop strategies to try to counteract this dysbiosis and promote a healthier microbiota. So with the team, we developed two approaches. The first one is a natural approach, in which we will use a fruit as a prebiotic to favor beneficial microbes, and this fruit is a Kamikami. We are also developing a second approach that is a bit more complex with an antibiotic treatment and the fecal microbiota transplant in the Gutsy study. So to first describe the first approach, we will use Kamikami. So what is Kamikami? It's a small berry. It's found in Amazonian region in South America. Its consumption is authorized by Health Canada. It's recognized for its rich content in vitamin C and polyphenols. And polyphenols, that, you might have heard of them. They are beneficial products that promote health coming from different fruits. So I don't have time to go through what the research done with Kamikami. There's actually really few, but we know that in mice models, it can actually reduce inflammation. And in human smokers, if smokers drink juice for 7 days, they have reduced level of different markers of inflammation. And so in Quebec, our collaborators have shown that in mice, the effect, the anti-inflammatory effect is actually due to a modification of the gut microbiota and the promotion of beneficial microbes. So we developed the Kamikami study, which is a pilot trial 032, which will be the first trial assessing the effect of Kamikami in people living with HIV. So our hypothesis is that by consuming Camu extract in capsules, and we actually prefer capsules because the taste of Kamikami is supposed to be not supposed to be very good, very tart. So we think that by consuming Kamikami extract, the gut microbiota composition will improve and people will have more beneficial microbes, and it will then reduce several markers of leaky gut and inflammation. So we developed a single-arm, non-randomized open-label study in which we will recruit 22 ART-treated participants, all adults living with HIV, with a CD4 to CD8 ratio below 1 to target those that have higher risk of developing non-AIDS comorbidities and have higher risk of inflammation. So people will continue taking the antiretroviral therapy during the study. And after screening in 2 baselines, they will take Kamikami capsules daily for 12 weeks and then stop for 8 weeks and be asked to come for a last visit 8 weeks later to assess the carryover effect. We will collect blood at each visit to assess the level of different inflammation markers. We collect stools also to assess the composition of the gut microbiota. And in an optional sub-study, we will ask participants to give colon biopsies taken by colonoscopy before and after treatment. So to quickly go through our different objectives, we will assess the effect of Kamikami on the reduction of several markers of the leaky gut and inflammation in the blood. We will assess safety and tolerability in people living with HIV and antiretroviral therapy. We will assess the effect on the gut microbiota and also on HIV reservoir size. So HIV reservoir are those cells that persist, those dormant cells infected with HIV that persist even after years of course. And thanks to 2 baselines, we will be able to assess the intra-participant variability of our parameters. In exploratory objective, we will use new techniques to measure the reservoir and try to use also the latest markers of gut damage, leaky gut, and inflammation. And in the sub-study objective, so in the gut mucosa, we will look the direct effect on the leaky gut, on the gut mucosa architecture, and in cell activation in biopsies. So this study is developed with the support of the CTN, and thanks to Judy Needham and John McBain from the CTN, we developed the protocol, the investigator, and the investigator brochure and the informed consent forms. So we also included in the investigator brochure product containing vitamin C, because there's really few studies assessing Kamikami. Dana Noinek from the CTN submitted those documents to Health Canada, and we received approval last November. We then rapidly submitted our study with our team to our local IRB in Montreal, and we received approval. In the meantime, William Shaw from the CTN developed the INFORM database to collect the case report forms. And this database has been said it needs to be put in production soon. Earlier this month, we organized a starting visit in our site. And actually this week, Judy Needham from the CTN issued the letter of authorization and the drug release. We are now ready to recruit. So to quickly discuss the second study, so we will use fecal microbiota transplants. So the goal is to actually take fecal product from a healthy donor and to transfer them in a recipient that is known to have a dysbiosis, so problem in the microbiota composition. And so we know that this technique can actually promote a healthier microbiota, beneficial microbes, and can reduce inflammation. So there's several ways to perform FMT, fecal microbiota transplant, with colonoscopy or gastroscopy, but we will use a new technique that is less invasive with gelatin capsules, those blue capsules here, that have 3 layers and open directly in the small intestine of the participant. Those capsules will be prepared by Dr. Michael Silverman's teams in Ontario and have a lot of experience with FMT in infectious diseases and also other diseases such as IBDs. So we really think it's important to assess FMT in untreated people living with HIV because this first results from a randomized study in Spain showing that it can reduce markers of gut damage such as IFAB, so it can reduce the leaky gut. But the study didn't use an antibiotic treatment before FMT, and we think it's really important to use this antibiotic treatment to get rid of the participant microbiota and favor the transplant of the donor microbiota. So our hypothesis is that with fecal microbiota transplant, we will counteract dysbiosis, promote a healthier microbiota with beneficial microbes, and once again reduce markers of the leaky gut and inflammation of treated participants. So to go quickly, it will be a randomized double-arm single-blind studies in which we will recruit 20 treated participants, once again with a CD4 to CD8 ratio below 1 in Montreal and in London. In the treated arms, people will take the ART throughout the study. We receive an antibiotic treatment for 5 days in the bowel clinic, and then we receive FMT capsules at day 0 and 22, and be followed for 12 weeks. In the placebo arm, people will not receive the antibiotic but a placebo, and will also be will receive placebo capsules at day 0 and 22. Once again, we will collect blood and stools at each visit to assess markers of inflammation and gut microbiota composition. And I don't have time to go through this objective. The study is actually in development, and we are actually confirming that objective soon, and I hope to present an update in the next meeting. So I would like to thank all our team, our members of our clinical CBIS, and our collaborators, and of course, all the study participants of the past and the future studies for our support, for their support. And if you're interested in FMT, we published in Frontiers in Medicine in an open access journal this paper discussing the relevance and the challenges of performing FMT to counteract leaky gut. Thank you for the CTN for the support, and I'll be happy to answer any question now or later by email. Thank you.